This video is to explain about light-dependent reaction. Photosynthesis basically have two different reactions. There are two stages in photosynthesis that includes first light-dependent reaction, which require the light energy, and the second one is the light-independent reaction, which does not require light energy. Light-dependent reaction occurs in the thylakoid membrane, while light-independent reaction occurs in the stroma of the chloroplast. As you can see from the diagram here, light-dependent reaction occurs in the thylakoid. The energy is absorbed in the thylakoid. H2O is used and then producing oxygen and ATP and also an ADPH. While for light independent reaction, ATP and an ADPH from the thylakoid membrane or from the light dependent reaction will be used to convert carbon dioxide to sugar. First, you need to understand about photosynthetic pigments. Photosynthetic pigments is to absorb light energy. So different photosynthetic pigments will absorb different wavelengths of light. The light energy is also known as photon. There are two types of photosynthetic pigments. The primary pigments, which are the chlorophyll A, and the accessory pigments, which includes chlorophyll A, chlorophyll B, and carotenoids. Chlorophyll A, there are a few types of chlorophyll A. The primary pigments that involved during light dependent is chlorophyll A P680 and chlorophyll A P700. P680 means the chlorophyll A absorb light energy at wavelength 680, while for P700, the wavelength that absorbed by the chlorophyll A is 700 nanometer. Accessory pigments include chlorophyll A except for the P680 and P700. And then we also have the chlorophyll B, the blue-green pigments, and also carotenoids that includes carotene, xanthophyll, and phaeophytene. The important component during light-dependent reaction is the photosystem. A photosystem consists of molecules of reaction center, which includes the chlorophyll and primary electron acceptor. So the chlorophyll here means the P700 and the P680. When the primary electron acceptor accept the electron, then it is said to be reduced. The second component of a photosystem is light harvesting complex, which includes the accessory pigments molecule. The chlorophyll A, except for the P680, the chlorophyll B, and also all carotenoids. So based on the chlorophyll A, there are two photosystems. The photosystem 1 that which is the chlorophyll A P700, while photosystem 2 is the chlorophyll A P680. Light-dependent reaction occurs in thylakoid membrane. It requires light energy, which is called photon. The light energy will be absorbed by the photosynthetic pigments and then converted to chemical energy, which is ATP and NADPH. There are two mechanisms of light-dependent reaction. The first one is non-cyclic photophosphorylation, and the second one is cyclic photophosphorylation. Non-cyclic photophosphorylation involve both photosystem 1 and photosystem 2, while cyclic photophosphorylation involve only photosystem 1. There are three stages for non-cyclic photophosphorylation. The first one is photoactivation. Photoactivation in the primary pigment. The electron in the primary pigment will be passed to the electron transport chain. The second one is photolysis of water, the splitting of water molecule. Then photophosphorylation, the synthesis of ATP during the transport of electron. Photoactivation occurs when the accessory pigments absorb the photon, the light energy from the sunlight, to the primary pigment. The primary pigment is in the reaction center. Then the electron in the primary pigment is raised to a higher energy level. They become more reactive. Then the electron from the reaction center will be excited and then released to the primary electron acceptor. So this is still in the reaction center. The excitement of the electron to the primary electron acceptor causing an electron deficiency in the photosystem. It becomes a bit more unstable because they lost electron. As you can see here, the energy, the photon, is absorbed by the accessory pigment, the green molecule in this diagram. Then the energy is channeled to the primary pigment in the reaction center. So the reaction center becomes more energetic, more reactive, and electron is then photoactivated to be passed or to be excited and pass to the reaction center. Pass to the primary electron acceptor. So electron from primary electron acceptor will then be transported through a series of electron transport chain. So bear in mind, the primary pigment has already lost an electron. Okay, this creates a deficiency in the primary pigment. So the electron needs to be replaced. Photolysis of water is important to replace this electron. This involve only photolysis of water only occurs in photosystem 2. The process of splitting of water molecules by light energy or the photon. Because we know that in a molecule of water, it is composed of H2 and hydrogen and oxygen. So the bond between the hydrogen and oxygen is covalent bond, which is very strong. So the energy from the photon will help to break the bond between hydrogen and oxygen. Release hydrogen that composed of proton and the electron. The electron from the hydrogen will replace the lost excited electron from the PS2. While hydrogen ion, the proton, will be released into the thylakoid space. And the oxygen will be forming oxygen gas and then released into the air. The third stage of light-dependent reaction is photophosphorylation. It is a process that synthesizes ATP, and the energy used to synthesize ATP comes from the transport of electron. There are two types of photophosphorylation, there are two types of electron flow. The first one is non-cyclic photophosphorylation, and the second one is cyclic photophosphorylation. Non-cyclic photophosphorylation involve PS1 and PS2, while cyclic photophosphorylation involve only PS1. Let me explain about non-cyclic photophosphorylation. The first step in non-cyclic photophosphorylation is the absorption of photon by the accessory pigments of the PS2. So to explain non-cyclic photophosphorylation, we start the explanation with photosystem 2. The photon that is absorbed by the accessory pigment will be transferred from one accessory pigment to another until it reaches the primary pigment in the reaction center of photosystem 2. So when P680, the PS2, undergo photoactivation, the electron in the P680 will be raised to a higher energy level and become excited. So the electron will be photoexcited and pass to primary electron acceptor. The excitement of the electron from the P680 to the primary electron acceptor will create an electron deficiency in photosystem 2. Thus, the electron lost in the P160 P680 will be replaced by electron from photolysis of water. So during photolysis of water, water will be split into hydrogen ion, oxygen, and electron. And once the electron is replaced, the P680 will return to its reduced state. From the splitting of water, the oxygen atom will combine with another oxygen and form oxygen gas and release into air, while the hydrogen ion, the proton, will be released into the thylakoid space. The electron will replace the lost excited electron from the P680. Then, the photo-excited electron that are passed from the primary pigment, the P680, to the primary electron acceptor will be passed through a series of electron transport chain. The electron transport chain consists of plastoquinone, cytochrome complex, and also plastocyanin. So the electron is actually passed from photosystem 2 to photosystem 1. The electron transport chain in this topic is the concept is similar with what we have learned in cellular respiration. Only the components is a bit different. Instead of succinate, dehydrogenate, and everything, we in this electron transport chain, it involves plastoquinone. In cellular respiration, we have ubiquinone, right? So in plants, we have plastoquinone, cytochrome complex, and the plastocyanin. You have to memorize these components. The transfer of electrons through the ETC provides energy to synthesis ATP from ADP and inorganic phosphate, while the hydrogen ions are pumped to the thylakoid space and creates proton gradient that will be used in chemiosmosis. This is similar what we have learned ETC and chemiosmosis in cellular respiration previous topic. Let's explain using this diagram. So the first step is photoactivation. The absorption of the photon, the light energy is absorbed by the accessory pigment. Consequently atau simultaneously in photosystem 1 and photosystem 2. So the photoactivation is when the electron in the photosystem, the reaction center, is excited to the primary electron acceptor. So the electron that comes from the P680 will be passed through a series of electron transport chain that consists of plastoquinone, cytochrome complex, and plastocyanin. And then the electron will be passed to the photosystem 1. And the electron, when it arrives the photosystem 1, it is actually replacing the loss of electron that occurs during photoactivation in photosystem 1. While the electron from photosystem 1 will be passed to the primary electron acceptor and then passed to a series of electron transport chain that consists of ferredoxin and ADP+. So in the end, in non-cyclic photophosphorylation, the final electron acceptor is the NADP+. The NADP+ will receive the electron and combine with the hydrogen ion from the thylakoid membrane and thus produce or converted or reduced to NADPH plus H+. At the same time, the photoactivation also occurs in the PS1. So this is a diagram showing what happens in the thylakoid membrane. This is the membrane. The pigments are embedded. The photosystem are embedded in the thylakoid membrane. The electron is passed from PS2 through through an ETC and arrive at the PS1, replacing the loss of electron in PS1. And then electron from PS1 will be passed to ferredoxin and NADP+. While the hydrogen ions that is released into the thylakoid space will be used to synthesize ATP through chemiosmosis process. The next is photoexcited electrons pass from the PS1 down a second electron transport chain through the protein pyridoxine and received by the NADP plus as final electron acceptor. The reduction of NADP plus is catalyzed by an enzyme named NADP plus reductase. So this enzyme transfer the electron from pyridoxine to NADP plus. So you have to memorize the name of this enzyme. The name is easy because the enzyme transfer the electron to the NADP. Thus, NADP will be reduced when it receives electron. Hence, the name of the enzyme is NADP plus reductase, which means it will reduce the NADP plus by transferring electron to the molecule. So from the electron comes from the P700, while the hydrogen ion comes from the thylakoid membrane. As a summary, the product of non-cyclic photophosphorylation on NADPH and ATP. So the NADPH plus H plus and ATP will be used in the stroma of chloroplast, which is in the Calvin cycle or Calvin cycle or the light independent reaction. There is another product from non-cyclic photophosphorylation. Let's not forget the oxygen. So oxygen gas will be released into the atmosphere. Cyclic photophosphorylation. So cyclic photophosphorylation, the word cyclic means berkitar kan. So it means that the electron, it's about the electron um, that is photoexcited from the photosystem 1 will return to photosystem 1 again. So there are 4 steps that can explain cyclic photophosphorylation. So first you have to remember that cyclic photophosphorylation only involve photosystem 1. Similar to what happened in non-cyclic photophosphorylation, the step 1 in cyclic photophosphorylation is when the photon is absorbed by the accessory pigments of PS1. Then the photon is transported or transferred to the primary pigment, the reaction center in the reaction center. Then the reaction center, the primary pigment becomes photoexcited and excite the electron to the primary electron acceptor. So it is said that P700 undergo photoactivation. The loss of electron from the PS1 creates an electron deficiency. The step 3 in cyclic photophosphorylation is when the electron is passed from the primary electron acceptor to the component of electron transport chain. The difference between this ETC from the non-cyclic photophosphorylation before, in cyclic photophosphorylation, the component of electron transport chain are ferrodoxin, cytochrome complex and plastocyanin. Okay, berbeza tadi plastoquinone, PQ. But then in this cyclic photophosphorylation, the component starts with ferrodoxin. So the photoexcited electron is passed from the primary electron acceptor through a series of electron transport chain, and the transfer of electrons will provide energy to synthesis ATP from ADP and inorganic phosphate. This is done through chemiosmosis process, as we have discussed in our previous chapter. Step 4 is when the electron from the plastocyanin return to P700. So the product of cyclic photophosphorylation is mainly, the only one is ATP. And this ATP will enter Kelvin cycle.